Mhm. Yeah. Hello, everyone. My name is Himanshi Singh. I'm a PMRF PhD candidate in IIT Kanpur PSB department. I hope all of you are doing really well and you guys are learning new things. And if there are any issues and problems with this current lecture, that is week 5 lecture, please feel free to share with me and I will try to resolve them. And if not, if I'm not able to resolve them right now, I'll make sure that in the next session, we'll clear those doubts. So, um, I hope that you guys must have gone through this week's lecture, which comprised of 5, I guess, video sessions, video lectures, and each one lecture was primarily dedicated for neuronal system, right? So in this week, if I want to brief you guys what we studied, it was basically about neurons, nervous system, right? The structure of neuron, how neurons are transporting signals, how we are sensing. If you remember in the past few sessions, we were discussing about sensing things, right? What is a thing which connect us from environment, right? Which connects environment to us, how we respond, how we change our behavior depending upon what kind of stimuli we are getting. So all those things, those are what to say, mega scale things, right? As if I take the example of fire, right? Fire or very some sharp object. So those are big things you can see with your naked eyes that it's there, and things like you can process that. If you can see it prior to stepping on a sharp object, obviously you won't step on it, right? And if you're not able to see that, then the moment you are stepping on it, there's a reflex action which allows you to react in a very short interval of time, ensuring that the damage is not too much to your system, correct? So this indicates the importance of sensing. And for sensing, you know that we have sense organs and all that. Those were macroscopic scale things, right? Which you can see. But what exactly is happening at a molecular level or at a cellular level, we don't know much about it. Though we know we have ample amount of information, but still we are lagging behind, and there's a lot of things which are yet to be discovered and yet to be analyzed or yet to be proven right. Correct? That is the beauty of biology, because the more you know, the more you don't know, right? Earlier, it was thought that only DNA can make proteins, right? If you guys remember that, from RNA to DNA, DNA to RNA, DNA to protein, sorry. That's it. But we know that this is not the sole case, right? There are RNA viruses which can make proteins and enzymes. So looking at biology field as a whole, we can say that the more we know, the more we don't know, obviously. But talking about sensory system or senses, which make us, right? Which make us conscious. Conscious is a very big term. At least I am not uh, sure what consciousness is. Like a very strict definition, I'm not sure about it. What comprises of consciousness, but I'm sure that consciousness does have a parameter of sensing, right? Because you are conscious. How you know that you are conscious? Because you are able to feel your environment, you are able to feel your surroundings, you are able to react depending upon your surrounding and changes in the surrounding, right? So this is one parameter which comprises of consciousness, but this is not the complete consciousness. So to understand those things, we should know what exactly is happening at a molecular level. From a macro level, we know that our skin, which is the outermost layer of our body, if it, because this is the thing which gonna sense stimuli. If we are talking about the extroception or the external stimuli from the surrounding, not from within, we are talking about from surrounding stimuli. If we are having stimuli from surrounding, so our skin will definitely respond to it. It should have sensors or something has to be there to make sure that we are connected with our environment, right? So even skin we can see, but what is happening in the skin, we don't know, because it is at a cellular level. And even at the cellular level, we can further go down inside single cell at a molecular level. What exactly is happening at a molecular incident the moment some external stimuli arrives or some external stimuli present itself? Correct? So for that, we should know the structure of a cell. You guys agree with me, right? If we know the structure of a cell, we don't know what is going on inside. But if we know what the structure of a cell, we can somehow speculate that if A, B, C, D, E, F, G things are available on the cell, they have this function, X, Y, Z function associated with these things. We can speculate that, okay, this function is associated with this product or this protein or this commodity of a cell or this part of a cell. So this function infers to sensing change in pH, right? Then we can say if in the external environment the pH is changing, we can say that this cells, this component will respond to the change in pH of the surrounding. Correct? So in a way, if we know the structure of a cell or say the structure of a functional unit of life, we can speculate some of the functions of it. Correct? Because via structure, we can relate to the function. We might not be always correct, but we will have some idea that what this particular aspect of a cell is associated with. Correct? And in a cell, I guess most of you, and I hope that every one of you knows the basic structure of a prokaryotic and a eukaryotic cell, or say plant cell or animal cell, right? So if we talk specifically about animal cell, so in animal cell, I'm talking about a very fundamental structure which is present in both prokaryotic as well as eukaryotic cell. It's a universal feature of a cell, that is a cell membrane. Correct? A cell membrane, is the outermost layer of a cell which make the cell, right? It compartmentalizes the cell from the external environment to the internal environment, just like our skin, which separate us from the external environment to our internal environment. Similarly, each cell has a cell membrane associated with it, which separates the cell's internal things or internal compartmentalization or internal organelles from the other cell or from the external environment or from the extracellular matrix. I hope that you guys have went through this week's lecture and you know what extracellular matrix, extracellular fluid, extracellular space is, right? So a cell has a universal feature of cell membrane. A cell membrane provides the cell its shape, its size, right? And also it helps in maintaining a constant homeostatic environment of that particular cell. You remember in the introductory sessions of this particular course, we talked about homeostasis. What is homeostasis? Homeostasis is basically maintaining the optimum functionality of, if we are talking about homeostasis of organism, then we'll say maintaining the optimal conditions for the organism's functioning. If we are talking about the homeostasis of a cell, then we can say maintaining the optimal conditions for the survival of the cell. And here by optimal, I mean all the parameters, for example, temperature, pressure, uh, say pH, say oxygen concentration, say carbon dioxide concentration, say other gaseous components concentration. All these are at their best level. And here best doesn't always means the maximum level, right? Because these criteria will change depending upon what criteria we are looking into. For example, if we are talking about aerobic microbes means these microbes survive only in the presence of oxygen, correct? So they will prefer maximum concentration of oxygen for their survival. But if I talk about facultative aerobes, or say, let's talk about anaerobic organisms, they grow well enough in the absence of oxygen. So one organism's optimal condition is not necessarily the optimal condition for the other organism. This is what I mean when I'm saying that it depends from criteria to criteria or the function to function which we are looking at. Correct? So talking about aerobic microorganisms, so for them, what will be the optimal conditions? The optimal conditions will be ideally to have the maximum oxygen concentration, which is uh, uh, chemically possible for them, because there is a certain threshold and limit, right? Even in the air, we have percentage of oxygen associated with the air. We are not in complete 100% oxygen environment, right? So the maximum possible percentage of oxygen in the air for that microbe will be its optimum condition for its homeostatic and survival and further thriving for that particular organism. Again, this, if I'm talking about a microorganism or say prokaryotic organism, it's a single cell organism. So whatever conditions we are talking about regarding homeostasis with respect to temperature, pressure, pH, uh, your oxygen or CO2 concentrations, this will directly mean the survival of the cell as well as the organism, because it's a single cell organism. Are you getting my point? Right? It's a single cell organism. So whatever the criteria or whatever the optimal conditions for a cell is for that particular uh, organism is exactly what the organism is looking for, or it's exactly for that particular organism also, because it's a single cell organism. But if we talk about multicellular organism, let's talk about us. We are multicellular organisms, right? So our whole body contains cells, correct? Every part of our body has cell. Every cell or different type of cells have different homeostatic conditions within one organism. So for prokaryotic, whatever the homeostatic parameters are for a single cell are directly related to the organism's survival, correct? But for multicellular organism, the homeostatic condition of one type of cell doesn't necessarily mean that the whole organism is in a optimal condition. I'll give you some time to process this and I'll repeat again. What I'm saying is that for a single cell organism or prokaryotic organism, whatever the homeostatic parameters or the optimal conditions are for a cell will directly replicate or directly can be directly speculated for the optimal growth of the organism, because that organism is only one celled organism, right? But for multicellular organism like us, ensuring homeostatic or optimal condition for one type of cell doesn't necessarily correlate to the overall optimal or homeostatic condition of the organism, or say humans or multicellular organisms. Correct? Am I making sense? Yeah. So what I mean when I say that you need optimal condition for a cell. Now imagine if I have a cell on my skin surface, right? It's a cell which is facing different environment. This cell is present in an environment, in the external environment, and my weather is changing throughout the day, right? Sometimes it's too hot, sometimes it's okay, sometimes the humidity is fine, sometimes the humidity is too much, right? So it is facing ever changing external environment. So the optimal condition for the cells which are present on the surface of my body are different from the conditions which are for the conditions of cells which are present inside my body. Let's say if I talk about the mucosa lining of my mouth, right, which is inside my mouth. My mouth internal lining are not facing the external environment most of the time, or they are not in direct relation with the direct in contact with the external environment. Their environment changes when I'm eating something, right? Again, if I'm eating acidic food, for example, if I'm at having orange, it's a citrus fruit, right? If I'm having orange, my cells local condition, local environment, for the cells which are present inside my mouth are acidic conditions, indicating that my cells need to adapt to this increased acidity. Acidity here is a parameter which can be related to pH, right? For example, same pH change, if I'm subjecting my external cells or my skin cells, chances are those same conditions might be deleterious for my skin cells. But those are not harmful for my cells which are lining my mouth, because my mouth cells are different from my skin cells. Hence, the optimal condition for my mouth cells, which are forming my internal of my mouth, are different from the conditions for which my skin cells are optimized. So within one organism, I have different optimal conditions or different homeostatic conditions or different parameters for which those particular type of cells are acclimatized. Correct? I hope that you guys are getting my point. So with this very brief introduction, I would like to say that every single type of cell in our body has different optimal conditions, but there is a wide range in which all those subtypes are fitted. I'll try to explain again. What I'm saying, though the conditions for which my skin cells are optimized are different from the conditions for which my internal mouth cells are optimized, but the range in which my skin cell and internal mouth squamous cells are adapted will be similar, meaning there is a broad range of pH, temperature, uh, oxygen, CO2, and all the other parameters for which we all are optimized. But within that optimized wide range, for example, if it is a bell-shaped curve, right? I guess I'll draw if I can. I'll try. Okay. Yeah. So if this is the broad range, if this is the broad range of temperature, pH, oxygen concentration, CO2 anything, any parameter you can take. For example, I'm taking pH as a parameter, right? pH or temperature or whatever. And here I'm taking for, say, number of cells we can take. Yeah. Just ignore my writing, not adapted for this number of, I'm extremely sorry. Yeah, number of cells, right? So if we say the maximum growth can be seen at a particular pH, let's say if this is a physiological pH of 6.5 or 6.7, right? At 6.5 and 6.7, most of our cells are okay, but again, our gastric cells won't be this. Our gastric cell will be optimal at acidic pH. But so hence our gastric cell might lie somewhere here, like this is the maximum growth for my gastric cells, right? Similar trend can be followed for every different type of cell. But again, the whole organism will fit in this graph, will fit in this range. So there can be different sections of it. For example, one particular type of cell is thriving in this range. Other particular type of cell is thriving in other range. Correct. But still that will be within this limit or range which we say, right? So, yeah, correct. So this is what I mean. Within us, within a multicellular organism, there will be different type of cells which have different optimal conditions or different homeostatic requirements. But overall, the organism will be having a particular range. Let me simplify this. For example, we feel good when we are in a temperature of about, say, 25 to 28 degrees Celsius, right? We are okay with it. Even in these conditions, some people might feel the need of fan, some people might not feel the need of fan. On the other hand, some people might still feel the need of AC, right? Or a cooler or air cooler. But 25 to 28 is a optimal temperature for us, correct? But we know that we have seen temperatures like 49 and 50, especially in North India. We have seen 48, 49 degrees Celsius also. So we feel uncomfortable in those high temperatures, but we are still surviving, right? How we are surviving? We are surviving by stimulating and changing our local environment. We put ACs, right? If the temperature is too high, we put AC there. Correct. So with that point, with taking that point into consideration, we can say that overall the organism has a range, but within the organism there are different ranges for different type of cells. Correct. So to ensure that these internal conditions are maintained for every single type of cells, we need compartmentalization. By compartmentalization, I mean, for example, if this is a room, right, and I want to have just have a corner which is colder in comparison to my whole room, let's say a refrigerator, right? I'm keeping a refrigerator in my house to ensure that there's one area in my house which is dedicated for cold stuff. Even if the outer temperature is 50 degrees Celsius, that particular compartment has temperature of 4 degrees Celsius. So that is only possible when my refrigerator has a very heavy body through which heat cannot transfer, right? Because if heat can transfer, then there is no sense in maintaining the internal temperature of in the refrigerator, because it is not possible, because the outer temperature is higher in comparison to the internal temperature. So that is why we need boundaries. Here comes the role of cell membrane, right? So what cell membrane's primary function is to isolate a cell from its external environment so that cell's internal environment can be regulated or can be maintained depending upon its requirement. As I've given you example of skin cell, which is facing the outer environment, and my mouth internal cells, squamous cells, which are facing my mouth interior, right? So there has to be a boundary associated with it. And that boundary is what we called a cell membrane. Correct? So what is a cell membrane? A cell membrane is nothing. It is just, wait, let me erase this. Yeah. So what is a cell membrane? A cell membrane is an outer boundary or a covering of a cell which forms the structure of a cell and helps in isolating cells' internal things from the external environment. And I hope now you understand why we need this compartmentalization or separation from our external environment, right? Because every cell in multicellular organism is dedicated for particular role and function. Hence, if all the cells in our body are in the same conditions, then cellular processes might not occur as efficiently as they do. Correct? Because there are certain enzymes or certain proteins which require, for example, a high acidic condition or say high basic condition. So if all the cells are optimized at very, let's say, very basic conditions, that they require basic conditions to survive, every single cell from of a multicellular organism. So what will happen? Other processes which require not so basic condition will not function to their maximum, or they might not work at all, right? This is the basic reason why the cell membrane exists. If you go back to the evolutionary history of how a cell came into play, how a cell was formed, you will know that the primary goal of a cell membrane or of a cell, when we say that this is a cell, was only said when it was separated from the external environment, because then only life starts to begin. Why? Because we concentrated our functionality to a localized environment, ensuring that even if the external environment changes, we still have a small niche of place where we can do our work. Correct? Just like you guys must have your study room, right? And you have a TV room. So if you want to study, you'll study in your study room. You will study on your table and chair, because that is your place of work. That is where you work best. If you want to enjoy, if you want to have some free time, or if you want to talk to your parent, you go to the living room. Correct? Because living room is the place where you do these kind of activities. Even our house is segmented that in that way. Correct? Similarly, within the cell, there are places which require certain conditions for its functionality to be fulfilled. Even within the cell, I'm not even talking about the multicellular organism. In multicellular organism, we have different types of cells for which they have attained different functions. Even those different functions require suboptimal level of management. What I mean by suboptimal level of management, by this I mean, if we have a say a stomach cell, right? A cell which is directly in contact with the lumen of the stomach. By lumen of the stomach, I mean we are eating food, right? So it is passing through the pipe and it is getting into the stomach. Let's say this is the stomach. If this is the stomach, then the cells which are lining the lumen part of the stomach, they must have certain characteristics to ensure that they are able to survive in the stomach conditions or stomach lumen conditions. And what are the lumen conditions of stomach? It is highly acidic environment, because the moment we eat food, depending upon what we are eating, let's say if we are eating carbohydrates, fat or proteins, but as we have a balanced diet, most of the time when we are eating everything. We are not specifically eating protein, or we are not specifically eating carbohydrates. We're eating mixture of things, right? If you eat only like dal, chawal, roti, sabji, correct? Even these are mixture of carbohydrate, fat, your sugars and your proteins, correct? And your fiber, all those things. So everything is coming simultaneously and all together at once. So the cells which are lining the stomach, they need to be adapted enough, or their conditions have to be in such a way that they are able to survive in acidic conditions. But same thing is not true for our mouth cells, as I've earlier said. Though our cells are okay when we are eating citrus fruit, which are highly acidic in nature, but even that acidity is nothing in comparison to the acidity of a stomach. Imagine in our stomach, we have HCl acid. HCl is the strongest acid known. If you put HCl on your skin, you will burn. Why you are burning? Because the skin cells are not adapted for such high acidity. But on the other hand, since birth, we are eating food, right? We are eating different kinds of food, but our stomach are not burning on daily basis, right? At least in non-disease condition. There are conditions in which actually ulcers happen, and you know what ulcers are. Ulcers are basically your stomach internal lining are not able to sustain the acidity of the stomach, and hence they start rupturing, and the problem can further went to like cancerous states and all those disease conditions. Correct? I hope that you guys are now able to understand that what exactly is the importance of a cell membrane. Now, I hope that you guys know what cell membrane is made up of. we have been learning this since since our school days, that a cell membrane is composed of lipid bilayer, right? What is meant by lipid bilayer? So lipid bilayer means lipid bilayer. If we break down these words, indicating that our cell membrane is lipidic in nature. Lipidic means fat, and it's bilayer. Bi means two and layer means sheet, right? So our cell membrane is lipidic in nature and is made up of two layers of such lipids, correct? If we see in this representative diagram of a cell membrane, we can see that these, wait, let me get the laser pointer for you guys. Yeah, I hope you can see this. So if we talk about the cell membrane, we can clearly see that this is the first membrane or the top part of it. This is the exterior environment, indicating that this is the part of the cell membrane which is facing the outside of the environment, right? And this is the cytosolic part, indicating that this layer is facing the internal of a cell. And again, you need to keep this in mind that this is a patch of a cell membrane. A cell is a 3D structure, right? So if we are talking about spherical shape, then imagine a ball. And if we take small patch from the ball, this is what we are representing here, correct? So the upper layer is facing the exterior environment and the lower layer is facing the internal environment or the cell's interior things. Even though it is lipidic in nature or bilayer, the plasma is bilayer and lipidic in nature, the upper layer, that is the one which is facing the exterior environment, and the lower leaflet, which is facing the internal environment of a cell, have characteristic differences. Just imagine the type of asymmetry which we have. First, we are multicellular organisms. We have different types of cells, correct? Those different type of cells are made up of cell membrane. Every cell membrane has two layers associated with it. Those two layers themselves are different depending upon whether the layer is facing the exterior environment or the layer is facing the internal environment. Correct? Am I making sense? Right. So the level of asymmetry and the level of optimization at a cellular level, you can see that from the cell itself, the dissimilarities arise. Even a cell is not same outside part as well as in comparison to the inside. If we look in details, though I'm not going to get into too much detail, but these are some generic things which are present in a cell membrane. And I hope that you guys understood why we need to know this information, because we have spent enough time in understanding that why we need cell membrane, correct? So looking at this, we can say what we can see here in this representative figure. Let's decode one by one. We'll move from left side to right side. Okay. It's a cell membrane. You can see that this is a cell. This is the nucleus of a cell. And we are just zooming in into one patch of a cell. Correct? It's a 2D representation. Obviously, it will be in the 3D dimension. Now, outer layer, internal layer. They both have these representative structures, which are called lipids, or they belong to a special class of lipid, which are called phospholipids. Why phospholipids? Because they have phosphate group attached to this blob you are looking here, this circular part. If you zoom in into the circular part to its molecular structure, I am not putting it here because that was not the goal of this particular part of it. I just want you guys to have some basic understanding and basic, you should know what basic components are associated with the cell membrane, because at the end of the day, we are learning things which are happening inside the cell, right? So before knowing what things are happening, we should know its structure. That then only we'll be able to relate to its structure function relationship, correct? So this round part, or this is called the head part or the head group part. And these tails which you are looking here, these are called hydrophobic fatty acyl chains, or just don't worry about the terminology, just the fatty acid chains. Okay. So this head group part has phosphate associated with it. That is why they are called phospholipids, phosphate containing lipids. Okay. And the upper leaflet, and again, there are different types of phospholipids. Correct? So the phospholipids which are present on the upper leaflet of a membrane are different from the phospholipids which are present in the lower leaflet of the membrane. Right? This we have established when we started discussing about plasma membrane, that they are asymmetric in nature. So from where this asymmetry is arising? This asymmetry is arising from the components from which the upper leaflet and the lower leaflet are made up of. The phospholipids which are predominantly present in the upper leaflet are not necessarily be dominant in the lower leaflet. Okay. So we have cleared one component of a lipidic bilayer, that is lipid itself. Now, if we look at this pink color thing, this pink color thing is a protein, correct? Indicating that in the lipidic bilayer or the lipidic plasma membrane, we have proteins inside them, right? We can see multiple proteins here, right? We will talk about them one by one. You can see, right? There are multiple proteins associated with the lipid membrane, indicating that our cell membrane does not only contain, doesn't only have the lipid, but there are proteins associated with the lipid bilayer. And these proteins have different characteristics depending upon which type of cell we are talking about. Okay. So if it's a membrane of a neuronal cell or neuron, right? So they will have particular types of proteins associated with those, with that lipid bilayer of a neuronal cell. If you are talking about a cell from epithelials or type of epithelial cell, those membrane will have particular type of protein associated with the lipid membrane. Correct? Then only we know that this is an epithelial cell. How we are saying that this is epithelial cell? Because it has different characteristics. Every cell has different characteristics associated with it. If I want to further simplify it, for example, if I ask you, are you? Right? So you're not gonna say Homo sapien, because every single individual right now today are Homo sapiens, right? Which means there are certain characteristics which are only true for you. So what are those characteristics? Those characteristics can be your name, your occupation, your place of living, or anything, any XYZ characters, like your physicality, correct? Anything can be there. So those are the characteristics which makes you you. And hence people around you recognize you based on those characteristics. Correct? Similarly, every cell has its own characteristic, and every surrounding cell recognizes other cell based upon these characteristic features. Correct? But every cell has characteristic features, but those features follow some universal trend or some universal type of things. Those type of things, for example, if I'm saying a protein is associated with the membrane, those protein can be of three different types. For example, if we can see here, first one is peripheral membrane protein, right? Other is integral membrane protein. Another one is lipid anchored protein, right? These are the three different type of proteins which we can see here, right? That this is peripheral membrane protein, this is a type of integral membrane protein, and this is a type of lipid anchored protein. So this is also a integral membrane protein. What do you mean by integral membrane protein? Integral membrane proteins, as the name suggests, integral means internal, right? So a protein which is transversing throughout the bilayer are called integral membrane proteins. Correct? What are peripheral membrane proteins? peripheral membrane proteins are the proteins which are associated with the periphery of a cell. That periphery can be both side, correct? It can be the upper leaflet or exterior leaflet's periphery, as well as the lower leaflet and internal leaflet's periphery. That is why you are saying peripheral membrane protein here also and a peripheral membrane protein here also. Correct? Yeah. So a protein which is transversing throughout the membrane, throughout the bilayer, is called integral membrane protein. A protein which is associated only with one side of a lipid bilayer, either the upper leaflet or the lower leaflet, is called peripheral membrane protein. Here you can see in the representative figure that these are proteins themselves, right? That these are proteins which are embedded in the lipids. But if we look at this lipid anchored protein, correct, we can see that the protein is here, but there is a different type of lipid. You can clearly see, right? There is this head group which we have discussed for these gray lipids, and then there are fatty acid chains which are this gray also, but they're different. This is a different type of lipid which is associated with the protein. Hence the protein is not directly connected to the lipidic bilayer, but is attached to a particular type of lipid which is part of the membrane, and hence this is called lipid anchored protein. Correct? Now there are different classes of integral membrane protein, peripheral membrane protein, lipid anchored protein, and they all serve different functions, obviously. So the specificity of a cell comes from the concentration as well as the type of proteins which are associated with the cell type, with that particular cell type. Correct? So the asymmetry or the specificity of a cell arises from different lipid composition in the upper leaflet and lower leaflet. That is the leaflet which is facing the extracellular matrix and the leaflet which is facing the internal of a cell, right? And also the asymmetry arises because of the type of proteins which are present in the cell membrane. Now, these different type of proteins, they can be either integral membrane protein, they can be peripheral membrane protein, and this peripheral membrane protein can be either associated with the upper leaflet of a membrane or the lower leaflet of the membrane. Correct? And then there is lipid anchored proteins also. Correct? I hope you guys now understand some basic structure of a membrane, and we can now move to our question session. So I can see Nikita is here. Nikita, if you're comfortable, you can unmute and just give a brief introduction about yourself, like your name, what are you pursuing, from where you are. And if you're not comfortable in doing this, you can just drop in the chat box. Yeah, it will help me to cater my lecture according to your background, and you might understand better.
Yes, ma'am. I am in third semester. I'm doing BSc Biotechnology honors. I'm from Shillong,
Okay. From where are you pursuing your BSc honors?
St. Edmund's College.
Okay, okay, okay. So is this your first session
No, no. But
with me?
like this Saturday, I'm joining for the first time.
Uh, With me, it's a first session, right? Have you attended my lectures earlier?
No, I have
Ah,
attended Tuesday's lecture.
okay, 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 okay. Got it, got it. Okay, so I hope you understood what, because as I can see, there are not many participants. So I hope that you can give me feedback.
Yeah.
So, so I hope that you understood. And are you facing any difficulties in the assignment or anything?
No, no, it's good.
It's fine.
After
Okay.
like attending the YouTube lectures, also it's easy to.
Okay,
So
okay, okay. And have you followed this current week 5 lecture completely? You done with the week 5 lectures?
uh, the one that's there in YouTube and Tuesday's lecture
Huh, okay, you are done with them, right?
and assignments too.
Okay, great, great, great. Going. Have you registered for the certification?
I'll do it by 2 days. There's like 4 days, I guess, remaining. So
Okay. So is this a requirement from your college, or you are
no,
have
it's
joined it
just
on your,
yeah.
sorry?
It's just like some of my friends, they decided that we'll be doing this course
Okay.
so that we can get this ABC credit.
Yeah. Because like, crediting is good, but I guess there are certain colleges which have requirement that you should fulfill your credits from NPTEL itself. Okay.
Yeah, those who are having this NEP.
Yeah. Correct. Okay, so we can go to the questions now, as you are the only participant. So I think we might end soon.
Okay.
Yeah. So yeah. So for today, what happened? Huh. So our first question for today, and yeah, feel free to ask any doubt or any question, and which is related to this week or not. I am happy to answer. Okay. Yeah, so our first question is which of the following ions has a higher concentration inside a cell as compared to outside? Is it potassium, sodium, chloride, or all of the above? I'll give you 1 minute because this is generally how I do things. I give 1 minute for each question and then we get the answer and some accessory information if needed. So your 1 minute starts now. You can drop your answer in the chat box, and this 1 minute is also for other people who are seeing this afterwards on YouTube, so they can also participate at the same time. Yeah, so your 1 minute starts now. Yeah, obviously the correct answer is potassium ion. That potassium ion is more inside the cell in comparison to outside. And if you have followed the lecture, do you remember what was the basis of this? That why this was intriguing? That sodium is more outside the cell and potassium is more inside the cell. Do you remember it? It has to do something with the evolution where the first life form arise, like sea, what ocean and all. It's okay if you don't remember. It was just a trivia thing, not a thing which you should remember. So obviously the sodium is more inside the cell, and this is a very generic distribution of uh, concentration of ions. But you should not take these numbers to the grave, as they vary from organism to organism as well as cell type to cell type. So we can just put it in some range in our mind that this is the millimolar concentration of sodium and this is the millimolar concentration of sodium outside the cell. So this might help you in just understanding what kind of conditions are present within the cell. Correct? So moving to the next question. So our next question is, the resting membrane potential of a typical neuron is minus 70 millivolt, minus 70 microvolt, minus 40 millivolt, or minus 40 microvolt. Your 1 minute time starts now. Yeah, obviously the answer is hmm. Yeah. So The answer is minus 70 millivolt. So would you like to explain this action potential figure? Would you like to attempt what you understood from this action potential graph? Feel free to feel free, whatever you understood, it's okay. Even if it is wrong, it's completely fine.
Okay, so it's like when the neurons they receive stimulus. So this there is
Let's start one by one.
depolarization.
Okay. So what is this part?
This is when this there it's in resting phase.
Correct,
Minus
correct, correct.
70 millivolt.
Hmm.
Yeah,
And then this is where we get the stimuli.
stimuli. Yeah.
Yeah. Then what happens?
Then depolarization happens. So the
Only if
potential
there is enough
enough.
threshold, right? Otherwise, not all stimulation will lead to depolarization. Correct? And
Yes.
what is that threshold? It's
It has to be more than minus 55
correct,
millivolts.
correct, correct. So if we have crossed the threshold, then only we'll get a
Depolarization.
response. Yeah, depolarization. After that,
After that, it reaches its maximum potential, which is plus 40 millivolts, and then it decreases again. I mean, then repolarization happens,
hmm,
and then this hyperpolarization.
yeah, and then again resting state and coming back to and getting ready for the next
Next,
input.
stimulus.
So in this figure, which part signifies that if in that duration another stimulus comes, it won't fire again? Which part? Is it the first part, the second part, the third part, the fourth part, or the fifth part? I'll repeat my question. I'm saying in this figure, there is a part during the cycle where if we get, for example, here we got the stimuli, correct? So before completing this cycle, there are chances that we can get another stimuli, right? For example, if I'm touching here, right? This is one stimuli. This is second stimuli. This is third stimuli, correct? But what if my speed is too fast, right? So chances are after a while, if you remember that if you are constantly touching at some place, sometimes it happens that you forget that you are touching, right? Because it becomes desensitized, correct? So in this graph, which region signifies that if in that duration another stimuli comes, we won't be able to see action potential, or in other way, we won't be able to sense the
Is it
stimuli.
4?
Yes, correct. Because from minus 70 to minus 55, we got the threshold crossed by putting our finger like this, correct? The action potential was there, though it's a very small, like we are working at a very, what to say, very large time scale, because I'm able to feel all the things which I'm doing with my hand right now, because this time duration is very less. You can see it's millisecond, correct? And my doing this is at least micro, or I don't think so, like at least in seconds, correct? So if another stimuli comes in this duration, meaning our first stimuli was there which crossed the threshold, it was repolarizing, and then if another response or another stimuli comes here, so you can see from, just imagine if this is minus, though it is not minus 80, let's say it's minus 90. Now the threshold is minus 55. So now if the stimulus is here, this hyperpolarization first needs to cross this threshold, correct? So earlier the threshold was this only, this gap, right? But now this is this much, correct? So if we are stimulating within one cycle of action potential, and if the cycle is in hyperpolarized state, we won't be able to generate any action potential or any signaling or any sensing, correct? I hope that it is clear to you. Moving to the next question. Yeah, so our next question states, resting membrane potential of a pacemaker cell is. I hope that you guys know what a pacemaker is and a pacemaker cell are. So the question states, resting membrane potential of a pacemaker cell is minus 70 millivolt, minus 70 microvolt, minus 40 millivolt, and minus 40 microvolts. And your time for a time starts now for 1 minute for this particular question. You can drop your answers in the chat box. Yeah. So the correct answer is, okay, I have one more. Okay. Yeah, so the correct answer is minus 40 millivolt. Minus 70 millivolt is for regular excitatory neuronal cells. Minus 40 millivolt is for pacemaker cell, because you need to remember what we can understand from here. Every detail is written in this figure. Feel free to go through this. I'm not gonna. It's similar to a normal neuronal cell, but one important factor which you should keep in mind, that why cardiac pacemaker cell do not have minus 70 millivolt, while all the neuronal cells have minus 70, minus 80 resting membrane potential, right? Imagine you, cardiac muscles are there, cardiac cells are there, pacemaker cells are there, right? So if the pacemaker cells resting membrane potential is minus 40 millivolt, correct, instead of minus 70 millivolt. So this somehow indicates you that for the pacemaker cells to get action potential from a very brief stimulus is easier in comparison to the regular neuronal cells. Correct? I'm repeating myself. I'm saying pacemaker cells having resting membrane potential of minus 40 millivolts indicate that they are more sensitive towards the stimulus, right? Because they are already somehow at the threshold level. So they don't need to wait for the signal to reach the threshold level. And imagine if that is the case, they are present in our heart. And if our heart is not generating enough stimuli, so it will stop beating, right? So to minimize and mitigate this, what if we have cells whose action, whose resting potential is already somehow near to the threshold level, indicating that all the stimulus, or say most of the stimulus, will get converted to action potential, in comparison to neuronal cell, where most of the stimuli gets converted into action potential if it crosses the threshold. So there is a lagging period. There's a lagging period for the stimuli to be effective enough to cross the threshold level. But if a resting membrane potential is already at a threshold level, chances are that all of our stimuli will result in action potential. And this will ensure that our heart is beating constantly with a regular interval of time, with a regular your curve or beats or whatever we want to understand from this particular thing. But here we are talking about action potential. Action potential at the end, at macro scale, what it will lead to, at the end, it will lead to the functionality. The functionality of a heart is to beat at regular interval of time. That regular interval of time, signal needs to be there, correct? And if that signal is there and our heart is waiting for the threshold level, our beating time will increase, right? Our stimulus is here. Our heart is waiting that the stimulus should be strong enough that it should cross the threshold, then only it will beat. Correct? Then this will ensure that our heart is not working effectively. On the other hand, if my stimulus is weak enough, but that weak has already crossed the threshold, which means our heart will beat at a regular interval of time, even translating all the stimuli into action potential. Okay. Our next question, our next question is, a negative membrane potential means there are blank outside the cell than blank inside. You need to fill in the blanks. So a negative membrane potential means there are more sodium ions outside the cell than potassium ions inside, or a negative membrane potential means there are fewer sodium ions outside the cell than potassium ions inside, or both A and B options for these blanks, or none of the options for these blanks. Your 1 minute for this particular question starts now. Feel free to answer even if it is wrong. Don't worry about it. Yeah, great going, guys. See, trying to attempt the question is the half battle won, right? Because when you are even attempting and trying to attempt the question, indicating that you are remembering things and trying to apply that, even if it is wrong, because if you have applied it, indicating that you have made certain decision and assumptions about it, and if it is wrong, we will correct it so that you will remember it better. And if you are right, obviously those signals will be stronger. The same thing works. We are studying our nervous system, right? We are studying neurons, and in the throughout the lectures you might have seen that a signal passes from one neuron to another neuron, right? Which was called synapse. From one neuron to another neuron, a signal was passing through this gap. So if you think continuously, thinking means what? You are passing information from one neuron to the other neuron, correct? Because you have learned something, and even in learning, you are getting information from outside to inside via your eyes. Your eyes get connected further to the brain. In brain, we have neurons, and neurons, these things are happening. So the more you rerun those things, the more stronger the connection becomes between those two neurons, which are associated with information transfer. Correct? That is why I always say attempt, even if it is wrong, because you're making connection stronger. So if you have made this connection which is wrong, because this is not the right answer, so what will happen? We'll correct it. So this neuron which was passing information to this neuron, which is a wrong information, right? But you have used this pathway. Now we have corrected it. So it will make newer connection now. So now the correct information pathway will be stronger in comparison to the older pathway. I hope I didn't confuse you guys much. But if you are not able to understand, feel free to ask questions. Okay. So the right answer is more sodium ions outside and potassium ions. Both of you, I guess you couldn't understand the question well. It's not a difficult question, but it's okay. The question was basically asking you what is the concentration of sodium and potassium outside and inside of the cell. In the previous question, we saw this table, right? So we can see sodium is more outside the cell, correct? Potassium is more inside the cell. Correct? So that was the whole soul question. What was the question? The question stating a negative membrane potential means, negative means this is the resting membrane potential. Our resting membrane potential is, as we can see here, is minus 70 millivolt. Correct? This is the resting state. This is how our neuron cell is in rest when they are not functional, when they are not transmitting information or signal. So in that resting membrane condition, what is the criteria for the membrane to be in resting membrane condition? That the internal is negative. And what is making the internal condition negative and outer condition positive? It is more sodium outside and more potassium inside. I hope you guys understood the question. It was not a difficult question. It's just it is how it was framed. Instead of directly using resting membrane potential, the terms were used as negative membrane potential. But you should know that even the resting membrane potential is in the negative range. It is the negative membrane potential. Okay. Yeah. Coming back to our next question. Sorry. So, which of the following is or are neurotransmitters? Is it GABA, acetylcholine, glycine, or all of the above? And your 1 minute starts now for this particular question. Again, feel free to ask questions and feel free to give wrong answers. We'll correct them. Okay. Well, our 1 minute is over for this particular question. So the right answer is all of the above. Obviously, if you have followed the week's lecture, you know that these all are the types of neurotransmitters. And depending upon the type of neurotransmitter, we name the neurotransmitters, right? If it is acetylcholine neurotransmitter, and it's a neuron which is associated in acetylcholine, we will call it cholinergic receptors or cholinergic neurons, correct? So the name of a neurotransmitter is what we name the neuron also, depending upon the neurotransmitter in which it is associated. So here is a very brief uh, representation of a nerve cell or a neuron and what exactly is at the synapse or between the two neurons where the neurotransmitter transfers, right? So what exactly are neurotransmitters? Neurotransmitters are chemical messengers that your body can't function without. Their job is to carry chemical signals or messages or information, which I am saying here, from one neuron to the next target cell. The next target cell can be another nerve cell or a muscle or even a gland. Okay, so this is our first neurotransmitter. This is our second neurotransmitter. So if, for example, this is a acetylcholine neurotransmitter, so it will release acetylcholine into the synaptic cleft. There will be acetylcholine receptors on this another neuron, and then they will identify acetylcholine, then they will do further signaling and all those, whatever the function was meant for that particular neurotransmitter. Okay. Yeah. So our next question for today is, yeah. So which of the following is not a property of action potential? Okay. So first is all or none. I hope you understand by all or none. This all or none doesn't mean these options with respect to option. It means all or none means all for example, if action potential is there, it will be the complete action potential or nothing at all. Okay, it's a 0 or 1. Either the signal is there or the signal is not there. Cool. Second option is it does not require a threshold potential to generate an action potential. Third is the amplitude of action potential is independent of the amount of current. Fourth is the frequency of action potential is correlated to the intensity of the stimulating signal. I'll give you guys 1 minute for this question. Feel free to properly read this question first thing, analyze all the options, try to understand what they are trying to say, and then attempt. Even if you haven't been through this week's lecture, in the past few slides, we have discussed most of the properties which are associated with action potential. Okay. So you have 1 minute for this. For those who are joining late, I hope you guys know that this particular session has been scheduled from 5 to 7. There is another session going on from 6 to 8, I guess. So just cross check in which session you guys wanted to be in, because we are already 1 hour inside this particular session. And the other session might have just started at 6 p.m. Okay. So the answer is it does not require a threshold potential to generate an action potential. For those who have given wrong answer, you guys understand where you have made the mistake. Just read the question first. The question states, which of the following is not a property of action potential? It is asking for the negative answer, not the thing which is the property of the action potential. Okay, so we have just seen in the last few question that for every action potential to be termed as action potential, we require a threshold potential. If the stimulus is not generating the potential equivalent, obviously more than the threshold potential, the action potential cannot be generated. And this statement says that it does not require a threshold potential, but this is the false statement, correct? We can clearly see here that this is the resting membrane potential. The first incidence is the stimuli. But if the stimuli is not enough or not more than the threshold itself, then there is failed initiation. Failed initiation of what? Failed initiation of the action potential. Meaning we do require, we does not know, we do require a threshold potential to generate an action potential. Okay? Yeah. So read the question properly. Okay? Also, I guess most of you answered third option, right? The third option says the amplitude of action potential is independent of the amount of current. So what this statement says, amplitude means this part. For example, if this is plus 40, right? This is plus 40 and this is the basal level, which was minus 70. So this is the amplitude, the maximum change from the basal level. So the statement is saying that this amplitude is independent of the amount of current, which is true, right? Because here, if the threshold is crossed, irrespective of what was the initial stimulus, the initial stimulus can be either of a magnitude of, for example, 0, or the magnitude of minus 40. Correct? So irrespective of that, at 0 also and at minus 40 also, the action potential will be generated at plus 40. Hence, the current, the initial current is actually not affecting the amplitude of the action potential. I hope you guys understood this. Okay, we'll go to the next question. So our next question states, the action potential in a neuron starts at dendrite, synapse, axon hillock, or nucleus. I'll give you guys 1 minute for this question and then we'll see the correct answer. Read the question properly and then answer it. All of you can attempt the question. It's okay, Narendra. I think it will be beneficial for you if you attend another live session which is going on at which started at 6, I guess. So you can attend that and you might gain more from that session because we are about to end. We had initial discussion. And also feel free to be in the session. It's completely fine. Anyone else wanna give a try to attempt this question? Feel free to attempt. I guess we have got answers from most of you guys. The correct answer is axon hillock. For those who have given nucleus answer or dendrite, I guess these are the only two answers. Synapse, no one said, which is good to see. As I said, read the question properly, right? So what the question says, the question is saying the action potential in a neuron starts at dendrites collect the information, they collect the stimuli, they aggregate the stimuli to make sure that the stimuli is above the threshold. The action potential is not starting from there. Correct? Then nucleus. Nucleus is the control center. There is no role of nucleus in action potential. If you remember how action potential is generated, it is solely based upon the change in the concentration of charged ions. There is no role of nucleus in it. The nucleus plays the role for the survival of the cell, not for the action potential part. Okay, this will get clear. We'll see a small video about it. But this is the very generic representation of the axon and how, from where the action potential starts. Feel free to read through this diagram, and also these slides will be available. I hope you all of you are able to access the session slides as well as the videos which I am uploading as soon as I can. And I'm sorry I'm not regular in it, but I'll make sure in the coming weeks I'll upload it within 1 or 2 days. So before moving to the further question, we'll see a small video about action potential, because this will then help you guys in actually seeing how the action potential is generated. So again, the same drill, you guys need to let me know whether you can see my slides and hear the video. Let me share my screen. I hope you guys can see the YouTube page. Just type yes. Any one of you can just type a yes in the chat box. Okay. And let me know if you can hear it also. I'll play it for a few seconds and then let me know if you can hear it. Just drop a yes or something in the chat box.
2-minute neuroscience, where I simplistically explain neuroscience.
Are you able to? Okay, cool. Then I'll play the video for you guys.
Topics in 2 minutes or less. In this installment, I will discuss the action potential. The action potential is a momentary reversal of membrane potential that is the basis for electrical signaling within neurons. If you're unfamiliar with membrane potential, you may want to watch my video on membrane potential before watching this video. The resting membrane potential of a neuron is around negative 70 millivolts. When neurotransmitters bind to receptors on the dendrites of a neuron, they can have an effect on the neuron known as depolarization. This means that they make the membrane potential less polarized or cause it to move closer to zero. This chart shows membrane potential on the y-axis and time on the x-axis. When neurotransmitters interacting with receptors causes repeated depolarization of the neuron, eventually the neuron reaches what is known as its threshold membrane potential. In a neuron with a membrane potential of negative 70 millivolts, this is generally around negative 55 millivolts. When threshold is reached, a large number of sodium channels open, allowing positively charged sodium ions into the cell. This causes massive depolarization of the neuron as the membrane potential reaches zero and then becomes positive. This is known as the rising phase of the action potential. The influx of positive ions creates the electrical signal known as the action potential, which then travels down the neuron. Eventually, the action potential reaches its peak. Sodium channels close and potassium channels open, which allows potassium to flow out of the cell. This loss of positive potassium ions promotes repolarization, which is known as the falling phase of the action potential. The neuron returns to resting membrane potential, but actually overshoots it, and the cell becomes hyperpolarized. During this phase, known as the refractory period, it is very difficult to cause the neuron to fire again. Eventually, the potassium channels close and the membrane returns to resting membrane potential, ready to be activated again. The signal generated by the action potential travels down the neuron and can cause release of neurotransmitter at the axon terminals to pass the signal to the next neuron.
You guys can see my slides now, right? Just yes or no in the chat box will help me. Anyone? You can see my slides now, correct? Cool. Okay, done. Right. So as we can see, that there has to be a accumulation of signal, then only the action potential can occur. So these are the dendrites, correct? So dendrites will receive signals. They will receive, and here by signal I mean current, electricity, or like charge distribution, all those things. Those signals will be transmitted here only when that threshold is available. Then only it will circulate throughout the axon, and this area is called axon hillock. So all the signals will get accumulated here, and if that accumulation is more than the threshold, then only it will be converted into action potential. Okay. So our next question is, information transfer through and between neurons is only chemical, only electrical, both chemical and electrical, or none of the above? You guys have 1 minute to for answering this question. I'm repeating again. Read the question properly. Good to see that all of you are replying. Okay, so I guess most of you have replied for this question. The answer is both chemical and electrical. For those who have corrected, I guess you got why I mentioned that read the question properly, correct? Because the question is asking about the information transfer through and between neurons, meaning within the neuron as well as in between the two neurons, right? So within the neuron, there will be electrical type of information, correct? Between two neurons, it will be chemical type of information transfer, correct? So the transfer of information from neuron to neuron takes place through the release of chemical substances into the space between the axon and the dendrites. These chemicals are called neurotransmitters, as you guys know now, and the process is called neurotransmission. The space between the axon and the dendrite is called the synapse, correct? Now, depending upon the type of neurotransmitter, we can name neurons, right? If it is associated with acetylcholine neurotransmitter, it will be called uh, cholinergic neurons. If it is associated with GABA, then they are like GABA associated neurons and all those different type of things. Now, coming the next question. Our next question is, which neurons are anatomically bipolar neurons? Spiral ganglion neurons, pyramidal neurons, dorsal cells, or Purkinje fibers? Your 1 minute for this particular question starts now. Even if you don't know, I guess you guys can attempt this question at least. But I hope that you remember this. It was covered in the session, if I remember correctly. Try to attempt even if it is wrong. Just try to eliminate the obvious choices and then look for the correct answer. Good to see that you all are attempting. Okay, the correct answer is spiral ganglion neurons. It's okay if you couldn't remember it, don't worry about it. So here is a representative structure of a neuron, which I guess you might now have very well learned. The amount of times I have shown you the neuron structure as well as how many times were discussed in the session. So I hope now that this basic neuron structure is imprinted in your mind. So as we can see, this is the structure of a neuron. You all know that these are the dendrites. This is the cell membrane, which we have spent enough time in the beginning of today's session to understand what is the role, what is the importance, what are the kind of things or everything associated with the membrane. So these are the dendrites. These are the cell body, which has like basically your nucleus in it. This was the beginning of a axon, which is called the axon hillock, where the action potential actually starts. Correct? This is the axon. This is your myelin sheet. If you have attended the week lecture, you know what is the role and function of this myelin sheath as well as oligodendrocytes. Correct? And the nodes of Ranvier, which is the empty space between two myelin sheath. And at the end, these are the axon terminal. And these axon terminal will end depending upon what type of neurons we are asking about. They will end either into the muscle, or they will be in contact with other neurons and all those things, and they'll be synaptic end of pulse, or they will be synapses if they are connected with other neurons. Correct? So this is a typical neuron. But here we are talking about further types of neurons. This is a basic structure of a neuron, but there are further types of neuron based upon their structure, dendrites, their spread of dendrites and all those. So if you see here, these are unipolar type of neurons, indicating that there's only one cell body, and then they have one polar site through which the information will be transmitted. Pyramidal is the one in which they are getting information from all the three directions. They are getting information from two directions, they're transmitting into one direction. Multipolar means they are receiving information, they have multiple dendrites spread all around them, and then they are transmitting the information. Bipolar means they have one set of dendrites, that is the information is coming from one side and it is uh, uh, eliminating, or it is transmitting to the other side. In Purkinje fiber, the one which they are getting information from like they are getting intense information and then they are transmitting the information towards one side. Okay. And if you don't remember, don't worry, go back to the lectures. The information is available in the lecture. Okay. So our last question for today is silver staining method that lead to exploration of neural anatomy was discovered by Raymond, Camillo, Ronald, or Carl von Frisch. I guess you guys can attempt this question. Yeah. So it's a memory-based question. It's not a logical question. So, but this staining is very important, and that is why I guess we should always remember those who paved the way for the field so that we can understand about ourselves with this intensity and intimacy, right? Yeah. So your 1 minute for this particular question starts now. Try to attempt. The correct answer is Camillo Golgi. Uh, those who have attempted, obviously, they are right. And for this, I have a very small video to actually uh, give you some hint that what exactly is silver staining and how it looks like. But before going to that video, so what exactly is a silver staining technique is, right? So a silver staining is used to detect proteins after electrophoretic separation on polyacrylamide gels. It combines excellent sensitivity in the low nanogram range whilst using very simple and cheap equipment and chemicals. It is compatible with downstream processing such as mass spectrometry analysis after protein digestion. The sequential phases of silver staining are protein fixation, then sensitization, then silver impregnation, and finally image development, right? And the image will look something like this. These are silver stained neurons. Just look at them, how beautiful they are looking, right? We have just seen the representative image of a neuron, right? Here, let's say this. This is the representative image of a neuron, correct? Now try to correlate it with the real structure of a neuron, right? You can see cell body here, correct? You can see dendrites here, correct? Then you can see axon, long axon here, through which the information can be transmitted. And you can see there are certain neurons which have very distant dendrites, correct? And then there are certain neurons which have very limited dendrites. Via this morphology only, we distinguish them based upon their structure, right? Also, I have given a very brief overview or a review article which is published. It's a good article if you want to understand silver staining. Okay, if we go back to this video, we can actually see what it is. So let me. Okay, yeah. So I might not show you guys this video because it's a big video. From in this particular video, they're starting from the history of silver staining to how actually it is performed. So feel free whenever you are free to give a try to this link. You might understand better what exactly silver staining is. And it is not only used for neurons, it is a general staining technique. It is used for staining proteins. How any of you have performed silver staining? Raise your hand if you have performed silver staining. Okay. It's okay. Have you performed? Okay, we won't go deep into it. It's a basic staining technique. So the principle is silver is a metallic ion, right? It's charged, it's positively charged. So if you put it on a protein, which is, for example, negatively charged, so what it will do? It will get bind to that protein, right? And silver can be easily oxidized. If you remember that people who wear silver jewelry, even in the environment, the jewelry becomes dark in color, right? It's because it gets oxidized. But if you are taking a silver at a like real and only silver in liquid form, then it won't get that color, the dull color which you get in the jewelry. It will give you brown color. So if you go to this video, you will get more information about silver staining. And also, this is not the only staining which is used for neuronal visualization. It is just a staining technique which is used for protein visualization. Okay, so if you want to visualize any protein, you can do silver staining. But before that, you need to do all the SDS page analysis, electrophoresis. So even if you are not familiar with these terminologies, don't worry. During your practical sessions, I hope that you get practical sessions in your colleges. So in your respective college, you will perform SDS page at one point or another point of time. Okay, so don't worry about the terminologies if you are scared about them and if you don't know them. These are basic laboratory practices to visualize protein, to check whether the protein is present or not, to see, and in further we can check whether the antibody works. It's a step-by-step process, so don't stress yourself much. Okay, so this is all from my side with respect to today's session. And as you know, I always pose a question by the end of every session so that you guys think about it, and in a hope that you guys develop a thinking habit about things which you have learned throughout the week. So for this week, we have this question. So question to ponder upon is, why nervous system has both excitable and non-excitable cells in place of only one type of cells? So if you have gone through the session, you know that even the neurotransmitters, there were different types of neurotransmitters, right? Acetylcholine, GABA, other like choline and all those things. So those were further divided into excitatory as well as inhibitory. So why we need both? Why can't we have only one type? Say we can have only excitatory type of neurons, correct? Or excitatory type of nervous system, and we don't have non-excitable cells, or say inhibitory cells. So what will happen? What will the consequence of having only one type of cells which respond to only one type of stimuli? So try to think on it, and you will definitely get the answer if you are listening to the lectures carefully. And if not, feel we can discuss it later, or you can just drop me a mail and we can have some discussion, and I might provide you, if it is available to me, I might provide you certain reading material to it, right? So for those who have joined late, I'm not going to repeat everything what we did, but just for a small summary point of view, we basically today focused on cell membrane. What is cell membrane? Why we need cell membrane? What was the role of cell membrane? How cell membrane came into play? What are the components of cell membrane? From where the heterogeneity in the response comes? Then the homeostasis of unicellular organism and multicellular organisms. And further, then we went with, as always, we went with our previous year assignment questions. We discussed certain properties and certain basic thing about neurons and neuronal system. So this is all from my side. If you have any question, feel free to ask. I'm here for, like, if you have question, I'm here, and if you don't have any question, I'm still available here in the next, for the next 5 minutes. Okay, and if you don't have any question, feel free to go. Thank you so much for your patience and attending the class. Hope to see you in the next session. Welcome all, welcome all. Thank you for attending the session. And if you have any issues pertaining to the course, or any doubt regarding the certification or exam, if I'm able to help, I will definitely help you all. Just you need to ask. I guess if we don't have any question, I'll end the meet here. Thank you once again.